Good afternoon, everyone, and thanks so much for joining us. My name is Mark Sabatine. I'm the immediate past director of the BRI, a cardiologist and a clinical trialist. And I have the privilege now of moderating this session entitled Where Are They Now? Brigham Research Institute Awardees. And this will be a chance really to spotlight some of the really amazing stars we have here at the Brigham, and note the support that they got earlier from the BRI and the great work that they've done, not only with that support, but how it's catalyzed future work. So each speaker will have about 10 minutes to present their findings, and then we'll have about 5 minutes for Q&A. And that will take us through this 45-minute session. I will note the session is being live captioned and recorded. So with that, looking here at the agenda, I think first up will be Vikram Khurana, who won the 2018 BRI Directors Transformative Award. And so the project title is N of Few, Targeted Clinical Trials for Neurodegenerative Disorders at Brigham and Women's. So Vic, we can't wait to hear about all the great work you've been doing.
Great. Thanks so much, Mark. Okay, terrific. Well, it's so good to be here, and I thank the BRI, the Brigham, for supporting what is a very ambitious project that would simply not have taken root without the support from BRI. So I have one relevant disclosure. Toward the end of my talk, I will mention a company, Humanity Therapeutics, that I helped co-found. Now, the BRI awarded me the transformative grant to really tackle a very terrible group of diseases, proteinopathies. As our specialized cells in our body get older, especially the ones that don't divide, like brain cells, neurons, we have a problem. Proteins accumulate and misfold, and we don't have good ways in our body beyond reproductive age of getting rid of them, and this leads to a devastating group of diseases. More than 50 million people worldwide suffer from Alzheimer's and Parkinson's disease, but not just these diseases, other diseases, rarer diseases of muscles, and some exceptionally common diseases like type 2 diabetes are all related to the misfolding of proteins as we age. And so this was really the problem that we set our sights on. And the fundamental question, of course, is why can we not prevent or modify the outcome for patients with, in our case, neurodegenerative disease? I'm a physician scientist, a movement disorders neurologist, and I'm particularly interested in this group of protein folding diseases. And our hypothesis is that a one-size-fits-all strategy may simply not work for these diseases, that diseases we know as one disease, like Parkinson's or Alzheimer's, are actually clusters of diseases, much as we've known in the cancer field for many years. And so to appropriately target a patient's disease, we would need to understand the biology of that patient's disease, and so develop precision medicine approaches for neurodegenerative diseases. And that doesn't just need good science, but it needs infrastructure, and that's where this grant has been so pivotal for me. I can confidently say that this award transformed my lab. I think we're transforming the Brigham, and I really hope we transform the outcomes for patients and their caregivers who suffer from these diseases. And this is the paradigm here, that patients could come into our clinic, that we could subtype them into groups on the basis of their genetics, biomarkers, and so forth, and that we could partner, whether with our academic colleagues or with industry partners, for therapeutics that might be now well suited to subtypes of patients, with the idea that a one-size-fits-all strategy may not work. And so our focus were diseases called alpha-synucleinopathies, which result from the misfolding of a protein called alpha-synuclein in the brain. The most common of these diseases is Parkinson's disease. And here you can see a brain cell, a dopamine neuron in Parkinson's disease with a pink circular structure here. That is aggregated abnormal protein that is leading to the dysfunction and the demise of these brain cells. And that's a cartoon picture of the alpha-synuclein protein that exists in many different shapes and forms in the cell, and particularly forms these fibrils, these fibers in brain cells that is associated with degeneration. Now, thanks to groundbreaking work 12 or 13 years ago from Shinya Yamanaka and colleagues, we can now take induced pluripotent stem cells, make these embryonic stem cell-like cells from skin of our patients, little skin biopsies, and we can turn those into brain cells, including the types of cells that are dysfunctional in neurodegenerative diseases. But the problem is that the advanced aggregation pathologies that we see in the aged brain simply don't occur in these immature brain cells that we make in the dish. And so we set out to tackle this problem because it's not only Parkinson's disease patients that suffer from this disease, but a whole family of other diseases, dementia with Lewy bodies, multiple system atrophy. And we think that in these different subtypes of the disease, there is a critical role for the misfolding, and in particular, the shape that this particular protein misfolds into. So if we're going to model this disease in the dish, from stem cells, we're going to need to have a handle on this particular advanced stage of pathology that we see in our patients. And so we set out to tackle this problem with a 2-year proposal, which was the focal point of our transformative grant. And so the idea was that at the time, our lab and others had been among the first to generate these iPSC models from patients with Parkinson's disease, but they weren't forming these aggregates. But could we develop a system where we could take spinal fluid, or from those patients who had passed their brain itself, extract these amyloid fibers, what we call PFFs, preformed fibrils, from this tissue or from spinal fluid in living patients, and then reintroduce that into brain cells from those patients that we were making in the lab. And this could then be used for genetic screens and drug discovery. And this would involve not just us, of course, but a number of collaborators at the Brigham, who you see listed here, but also building an infrastructure that links 3 different departments: neurology, pathology, and genetics. And that's where the transformative aspect came in. We needed to build an infrastructure. We also needed to work with the basic biology. So I'm not going to go into this slide in detail, but my lab has been mapping the molecular consequences of the misfolding of this protein for a number of years now. And could we take the genomes of our patients and start to interpret their genetics in light of all of the biology that my lab and others have developed around the misfolding of this protein and how it affects the cellular milieu in each of these brain cells. And so the idea was really to integrate genetic information as well into the way we model these diseases from our patients. And so in order to do this, we had to build clinical infrastructure. And in the Transformative Award, we focused largely on what we would do for biospecimens. How would we collect them and process them? And this involves sequencing the genomes of our patients. It involves taking skin biopsies and making stem cells. It involves taking spinal fluid and collecting brains of those patients who have passed, so that we can learn a lot more about this technology. And that in turn involved integrating 3 departments. And so some of the key successes in the last couple of years have been that we now have a genetics clinic in neurology. Many other divisions in neurology are following suit. And this is a clinic that is really full now at capacity. It's staffed by a genetics counselor and a NAGI, 2 clinical attendings, research assistants, nurse practitioner, and it has 2 fellowships, including a new Mass General Brigham neurogenetics fellowship. And so this is the clinical portal that enters our patients into this clinical and then scientific discovery process that we think will allow us to identify subtypes. We've made some great strides, I think, on genetics. We've worked with Shamil Sanayev at Brigham and HMS Genetics, at Jen Pang at the University of Illinois, to develop machine learning approaches so that we can mutationally profile our patients now and divide them into genetics clusters. This work's being led by a terrific postdoc who works with all of us, Sumaya Nazim. In order to have the infrastructure for brain banking, we needed to work closely with our pathology colleagues so that we could get brains, not just from Boston, but from New Hampshire or Maine, within a few hours of a patient passing away, so that we could bank those, flash freeze those brains and get the highest quality brains from which we could extract this toxic protein. 20 of these brains are now matched to stem cells, which is probably the largest collection we would guess in worldwide for these diseases. And so now we've been able to put this together, and Alain and Isabel in the group have been able to make the next generation of stem cell models for these diseases. Here you see an image of what we have typically done. We've met patients, we've made stem cells from their skin cells, what we call fibroblasts, and then we make the brain cells. We did this a number of years ago, as I mentioned. But now from the brain collection that we have, we can extract the synuclein, amplify it, and reintroduce that into the brain cells. And for the first time, we are seeing pathology in the dish that looks remarkably like, and so the dish is on the top, the cells we make, remarkably like the human brain, which you can see depicted below. So this is a very striking parallel that we have not been able to see before, and will give us new windows into the specifics of this disease in different patients. We use longitudinal live track imaging where we can watch these in real time, watch these aggregates form in brain cells, and we've now discovered that these are heterogeneous and that there are different populations and that they have different consequences on cell biology. We think these are really important insights for us to develop appropriate therapies. And finally, we're making much more complex systems. This grant from BRI has led to many other grants, including a shared $9 million grant with Lorenz Studer, who you see pictured there on the left, where we can now incorporate our neuronal models into the much more sophisticated three-dimensional and two-dimensional models that Lorenz's lab has pioneered. That work is now underway, would not have been possible without this award. And you can see here the many different groups from the NIH to private foundations that have now supported this work. And again, none of this would be possible without this being seeded by the BRI and this transformative award. And finally, on my last slide, we have actually begun this concept of N of few clinical trials. And by that, I mean, can we take patients where we understand the biology and can we design appropriate therapies for them, where we're not doing large-scale trials, but small-scale trials on a few patients where we really have taken a deep dive and we have a good understanding of their biology. I'll give you 3 examples that we're excited about. A young boy whose family is public about this, Tayson, has a terrible degenerative movement disorder. The Transformative Award has allowed us to team up with Tim Yu's group, who has designed an antisense oligonucleotide for this young boy, and Asha Koreka and Claudio Gusmao at the Brigham have been leading an effort to build stem cell models from this patient, to test the antisense oligonucleotide gene therapy in cells made from this patient to understand the on and off target effects of this. In other words, we'd like to predict not only the positive effects, but also the potential negative side effects of a therapy like this. And Claudio has been building the clinical trial infrastructure that will hopefully allow us to translate this into this young boy. We follow an incredibly interesting family in northern Spain that carries an alpha-synuclein mutation. Every generation, some of these patients miss the consequences of the disease, and we don't know why. And so the Transformative Award has allowed us to study these patients in detail. And remarkably, mouse models have been made for this mutation in our department by Dennis Selko, Silke Nuber, cellular models by Ulf Detmer. And we have found that a drug that is now in clinical trial has remarkable effect against this particular type of alpha-synuclein. And we are now planning a clinical trial on 3 young men in this family. And finally, a drug for a synuclein disease called multiple system atrophy recently failed in clinical trial because it did not meet its primary endpoints. We have been following patients with imaging with Tarun Singhal, Jeff Young, and other colleagues at the Brigham, work that's being led by Ariana Pitaro and Andy Willett in our group. And we've been following these patients over a couple of years now. And so we were in a perfect position to try a so-called failed drug in these patients. And with brain imaging, we've started to gain new insights that the drug is indeed engaging its target, but may engage it differently in different patients and even differently in different parts of the brain. And hopefully through these insights, we'll learn how to develop a next generation drug that is more effective. So none of this would be possible without the BRI and the Transformative Award. And I thank you so much for all of your support, and I look forward to updating you all in the future. Thank you.
Yeah, well, Vic, that was absolutely spectacular. I want to be mindful of time. Maybe just to note, just the key things for this. I'll just comment. One, diseases that are common. Probably most people, either through their families or through friends' families, have someone who has suffered from Parkinson's or Alzheimer's. So, amazingly, unfortunately, prevalent problem. The incredible collaboration and the notion that the inducible pluripotent stem cells, that model seemed incredible and is, but as you pointed out, was not quite enough, to be able to develop the therapeutics. And so it really is now incredible to take that to the next level. If people have questions, maybe put it into the chat. I do want to keep us on time, but thank you for sharing that. It's really spectacular. Next up will be Jean Lee, who got a 2018 Pilot Award. And so, Jean, if you want to start sharing your slides.
Yeah, okay. So in 2018, I was awarded a Pilot Award, which is really designed to provide funds for 1 year to support basic clinical or translational projects with the ultimate goal of supporting research so that the investigator can obtain preliminary data that's necessary to apply for new funding from granting agencies such as the NIH, which was our target. In the next slide, I'm going to show you some images of staphylococcal infections in humans. So if you're uneasy with looking at such photos, you may want to look away. Okay, so my laboratory studies the bacterial pathogen Staphylococcus aureus. And this organism causes a wide spectrum of human diseases, ranging from superficial skin lesions like boils and impetigo to toxin-mediated diseases such as toxic shock syndrome, food poisoning, or more life-threatening invasive diseases like bacteremia, endocarditis, pneumonia, and surgical wound infections. And this organism is a real problem because it's developed antibiotic resistance, as well as all attempts to develop a vaccine against this organism have failed. And in the corner here, you see an electron micrograph of Staph aureus cells. These are these coccoid-shaped bacteria, often referred to as MRSA or methicillin-resistant Staph aureus. Okay, so Staph aureus is able to produce such a wide spectrum of diseases because of all the different virulence factors that it produces. And these include surface proteins produced by the bacterium that allow it to colonize host tissues and adhere to a variety of different host cell types. And then it also secretes a variety of proteins like enzymes or toxins, proteases, nucleases, phospholipases, and these pore-forming toxins that actually kill many different types of cells, including the white blood cells, which are so critical for protection and against staphylococcal diseases by killing the bacteria that they engulf. And then also included in these toxins are the superantigens produced by Staph aureus. Okay, so in addition to all these different virulence factors, it was discovered only about 10 or 11 years ago that Staph aureus produces what are called extracellular vesicles. So again, here is this electron micrograph of Staph aureus, and you can see here secreting these nanoparticle-sized vesicles. And we can purify those in the laboratory from Staph aureus cultures, and you see them in their purified form here. They're enclosed by a membrane. And So this is not unique to Staph aureus because also mammalian cells, fungi, and other types of bacterial cells also generate these extracellular vesicles. But we thought that they probably play a role in staphylococcal diseases. So we took these purified extracellular vesicles and we performed mass spectrometry analysis on them to look at the different protein components, like what was their cargo. And it turns out that about 50% of the proteins that are inside these extracellular vesicles are actually cytosolic proteins, the soluble proteins that are usually within the bacterial cell, but are also enclosed in these vesicles, as well as membrane and cell wall associated proteins, these secreted proteins, and then a variety of proteins with an unknown function. In addition to the protein components of Staph aureus, we also found that these extracellular vesicles also contain Staph aureus capsular polysaccharides, as well as nucleic acids like DNA and RNA. So I showed you this slide before with the virulence factors of Staph aureus, but now I've highlighted in pink all of those virulence factors that are also contained within these extracellular vesicles, lots of surface proteins, enzymes, and a whole variety of toxins. And so that made us think that these vesicles probably did have a whole lot of biological activity and maybe played a role in the ability of Staph aureus to cause disease. So our hypothesis then was that these vesicles provided a secretory pathway for staph to transport their bacterial cargo in a concentrated form to host cells, so that rather than secreting them just in the environment, the vesicle contents that reaching the cells are protected from destruction by host proteases, nucleases, and toxin neutralizing antibodies. Specifically, we needed some preliminary data for our grant proposal to look at the cellular level at the interactions between these EVs and human macrophages, which is our cell choice. And so what we did in these preliminary data was we were able to demonstrate that human macrophages take up extracellular vesicles in a dynamin-dependent endocytic process. And you can see here, this is an electron micrograph of insides of a human macrophage. This is an endosome. And what you can see here are these extracellular vesicles that are decorated with these immunogold particles so that we can identify them. And then also we wanted to look at inflammasome activation and what the in vivo response to Staph aureus was. So this is our model of how Staph aureus EVs mediate inflammasome activation. And this is a process in which human macrophages respond to something such as a bacterial insult to the cell. And so in our model, we were able to ask a lot of questions, and with use of different bacterial mutants from which we purified EVs, we could show that the EV-associated lipoproteins that were associated with the bacteria, with the extracellular vesicle surface, interacted with the toll-like receptors that are present on the surface of human macrophages. This results in downstream signaling, NF-kappa B signaling, which ultimately results in the secretion of pro-inflammatory cytokines like TNF-alpha and IL-6. So this is the priming signal that's necessary for inflammasome activation. We were also then able, with other toxin mutants that we had, to show that these toxins contained within the EVs were essential for inflammasome activation because they mediated this potassium efflux, which is essential for assembling the NLRP3 inflammasome, activating caspase-1, which results in the proteolytic cleavage of these pro-inflammatory cytokines and causing their release from the cell of IL-1 beta and IL-18, which are very biologically active. We were able also for our preliminary data to show that if we injected mice with these purified EVs, they also responded to the infection by producing these inflammatory cytokines, IL-1 beta and IL-6, that we could actually measure in the peritoneal washes and in the serum of these animals. So, in summary of the data we were able to generate showed that EVs were indeed a novel secretory pathway by which Staph aureus could transport its cargo directly into host cells, and that EVs carry multiple virulence factors which induce cytotoxicity and death of those cells at a higher concentration, but at a lower concentration, stimulate an inflammatory response in these host cells. And these may play a role in the pathogenesis of staphylococcal infections. And what we're hoping is that a better understanding of the contribution of these EVs to disease pathogenesis may lead to better preventatives like vaccines or other new therapeutics to treat staphylococcal infections. So the return on the investment of the BRI into my laboratory was that, yes, we did get our R01 grant funded by the NIH to study the host pathogen interactions of EVs and macrophages. We had a couple manuscripts published, multiple presentations at local and national meetings. And more recently, we've been able to demonstrate that if mice are infected with Staph aureus bacterial cells, we can actually now demonstrate that they're generating these extracellular vesicles in vivo. We can purify them, we can quantify them. And based on some of these new data, we've actually now submitted 2 additional NIH grant applications, and we hope to better understand these vesicles and their role in staphylococcal and treatment of staph infections. Importantly, I wanted to acknowledge Xiaogong Wang, who's a junior faculty member in my lab, who actually initiated this whole EV project and has really driven it and performed the majority of the experiments with the assistance of Bill, Paul, and Olivia. And Diva Kara is our newest postdoc in the lab. He's a chemist, and he submitted one of the new investigator awards that were submitted recently. So with that, I'll end and take any questions. Thanks.
Great. Well, Jane, that's fabulous, and just a huge amount of work done with the pilot award. So kudos to you and your entire team for that. And certainly we would love for people to ask questions. I think Dr. Hata is on now. And I think if you haven't seen it, Jackie raised a good question. I think, one, you're obviously tackling a bug that is one of the most, if not the most common pathogen that, likely probably everyone watching has had some staph infection sometime in their life. Luckily, hopefully mild, but obviously, as you point out, they can be quite severe. In terms of sort of the future looking part for that, are there data for other types of bacteria, maybe gram negatives, where there's some data for extracellular vesicles, and does that appear to be a lever that people have been able to kind of pull on to try to thwart the bacteria? Sort of what's the long-term view in terms of how this could affect treatment?
Yeah, certainly the field with the gram negatives has progressed much earlier and faster than that with gram positive organisms. And there have been use of these extracellular vesicles produced by gram negatives as actually vaccine candidates, for example, against Neisseria meningitidis. Mm-hmm. We've also been exploring the vaccine potential of this, but it's just better understanding of, how these secreted molecules can travel a distance away and still exert their biological activity, not just at the surface of where the bacteria are, but again, against a distant cell.
Yeah. And then we have one question from Cynthia Lemire. I don't know if you can see it. I can read it, though, for people who can't. Complement is the body's first line of defense against pathogens. Do these EVs bind complement and then bind complement receptors on macrophages? Do they contain complement?
Yeah, that's really a very good question. We don't think they're actually that complement is essential for uptake by the macrophage of the EVs, because we don't have serum in our system right now that in those in vitro studies. I'm really not aware that anybody has looked at the role of complement and in relationship to extracellular vesicles? But this is a great question.
That's why we have these sessions. Okay, thank you so much, Jean. We'll now go to our final presentation. That was fabulous, much appreciated. And then Callum, if you want to start sharing then. All right, and the floor is yours, Callum.
So Mark, thank you very much for asking me to participate in this session. My original award was a Shark Tank Award in 2013, which seems like just about yesterday, called the Clinical Phenotype Innovation Pilot. One of the great and attractive features of this, and I think something that really highlights the importance of external funding for this type of work, is we did not need any preliminary data. And so I'm essentially a basic scientist, and I've worked for many years on the mechanisms of heart failure. But for this particular project, I took a totally different tack and was able to make a pitch for funding to really redefine how we measure diseases. So for most of the lay folks on the call, you probably recognize that many diseases have been around for a long time. With COVID arriving in the last year, you recognize that new diseases are coming out all the time. Most of the diseases that arise are not due to a new environmental factor, a new virus, but actually usually to a new test, a new way of measuring things. And in many instances, most of what we've done is really defined by collections of specific findings, patterns that are recognized to be associated with different outcomes. Example on the far right here is a condition that in the late 18th century was called dropsy. We haven't gotten much further than that now, although we do recognize that there are ways that we can, by measuring different blood tests, doing ultrasounds of the heart and other assays, we can at least differentiate whether this is due to liver failure, kidney failure, or heart failure. But in the 1800s, it was very difficult to make those distinctions. One of the core problems, however, is that even having done that, there's still further refinement that we could make, and many of the diseases that we currently have still have very diverse outcomes. And much of this variation is due to what we measure. We don't have enough tools in our toolbox to actually be able to distinguish the different causes or the different subsets of disease and then recognize those causes. And in fact, in many instances, the things that make diseases common are actually the things that make them mixed. They're the aggregation of different syndromes, different subsets of disease into one big cluster, because we only really have drugs for that one big cluster. And so this pilot was really designed to see if we could improve our understanding of disease by just measuring new things. And in the first instance, what we did was to take heart failure, and this was the actual focus of the pilot, and just to reclassify it based on things that typically don't get measured in heart failure. And I've shown a range of some of these things. We actually did over 30 of them. But one of them, for example, was just the way in which your little uvula at the back of your throat hangs down. So it turns out that the fusion of the top of your mouth, including this little area here, I don't even know what people call it in America, but we used to call it a gargle. And it basically is a soft tissue that protects the back of your throat, has lymphoid organelles in it. But ultimately, at the end of the day, the shape of it is actually tells you a lot about how your heart formed, because the same processes that cause your heart to form in the midline actually also cause the roof of your mouth to form. So it's a very powerful predictor of valve diseases and aortic disease that may contribute to heart failure. We also were able to identify, using machine learning, different patterns of retinal images taken actually with a smartphone that relate to the causes of your underlying heart disease. And then one of the other areas we explored was looking at the skin and the lining of your cheeks, a classical cheek swab, put it on a simple slide, look at it under a microscope, and we were able actually to see the abnormalities, the cellular abnormalities that were present in your right side of your heart, present in these cells from the inside of your mouth. We've done a whole host of electrical studies looking at conduction in the skin, conduction in your hearing apparatus, both bony and neural, and then even looking at your stride length as measured by a simple camera placed above the entrance to the echo lab, to be able to show that using these tools, you can define very discrete types of heart thickening, of inflammation or infiltration of the heart, and even decide fairly quickly whether something is inherited or non-inherited by the patterns with which you walk. And then finally, on the far right, simply adding a thermal camera allows you to discriminate different subsets of heart failure, because some of the forms of heart failure are actually associated with abnormalities in the way in which the lipid under the fat underneath your skin is distributed, and different types of fat have different heat characteristics. So we saw that this was actually a very powerful way of dissecting diseases, helped us identify some of the underlying genetics of these diseases. And so we began to think about ways that we could imagine finding completely new diseases, new tests, new assays, rather than taking tools from other areas and applying them in heart failure. And so for this, we were actually fortunate to be able to take the work that we had done for the Shark Tank and raise a considerable amount of money. This work has now raised over $120 million of additional funds, brought in a series of technologies, series of external entities, including Verily, the Google Life Sciences team, AstraZeneca, the American Heart Association, Apple, Quest Diagnostics, and several large pharmaceutical companies, and began to think about ways we could systematically build new measurements. And one of the ways that we first started to do it was to look at stress tests for molecules and cells. And so one of the difficulties with biology is understanding how everything fits together. And so one of the areas that's particularly inaccessible in most of disease, not so much in cancer, because you can take tissue out, you take the tumor out, or you can sample it from blood, but is cell information. And so what we did was to say, could the cells in your peripheral blood actually represent some of the mechanisms in your heart or in other parts of your body? And so what we did was to take those cells, add panels of perturbations, small molecules or physical challenges, stressing those cells in different ways. And you can see here in the middle diagram, here's a series of different cell types from peripheral blood. You add one small molecule and you see in a subset of patients, this completely new set of cells emerge. And what they are, this is actually based just on the shape and size of the cells, but you can do it for all sorts of different functions of the cells, and then use artificial intelligence to train those discrete outputs against particular outcomes that are important, such as being admitted with a particular condition in the subsequent 6 months or 12 months. And this is a particularly powerful set of tools for building a completely new portfolio of information about individual people and getting at mechanism at a single blood sample or from a single other test. And you can add perturbations. You can imagine, I'm sure anybody on the call can imagine lots of different combinations that you might use. So for example, we've used small molecules to look at calcium transients in cells in peripheral blood and identified novel blood clotting pathways that way. We've used AI plus exercise to look at classifying heart failure. Very limited amounts of intervention allow you to begin to identify completely new subsets of heart failure. And we've used a standard meal in the context of OCT, optical coherence tomography assay of your retinal capillaries, access into the blood vessels in your brain to begin to try and understand what are those factors that determine your risk of dementia, your risk of vascular disease, your risk of heart failure, from looking at accessible parts of your body in the context of a baseline and then a perturbation. The overall program was put together under a project that was named, not by me, I hasten to add, One Brave Idea, by the American Heart Association and Google Life Sciences. And what we did was really to try and use these technologies to begin not just to classify disease late in life when you've actually already developed symptoms, but to move way back into the earliest parts of life to try and identify signals by using these evoked pathways to identify signals before there was any evidence using conventional tools of any disease whatsoever. In our first pass, and we've done this only in a series of proof of concept areas and are now beginning to build the approaches, the funding, and potentially even spin out a company to try and do this at the scale that is necessary to do it across all of disease. But just for our cardiometabolic program, we've identified over 150 completely new genes that new phenotypes that are markers of diseases very, very early in life. So the outcomes seeded by this initial award are new tests for a whole host of different forms of cardiometabolic disease with different outcomes. By design, they're low cost, they're scalable, it can be deployed quickly. We built this around, for example, the traditional blood test that you get from when you go and visit your primary care doctor. But by adding some simple technologies plus machine learning, it's possible to deploy this across the whole country in a very efficient way once we actually commercialize the technologies. We've identified over 100 new human drug targets for a particular set of subsets of cardiometabolic disease. I'm sure many of these will fail, but the nice thing about it is we have human assays that allow us to reduce the risk and reduce the cost of drug discovery, because for every one of those steps in drug discovery, we can go back to the original humans that tested. We believe we're bringing research and clinical care closer together, finding new ways to match drugs or nutrition or environmental challenges to the individual. And importantly, and this is a slide on the right, looking at behaviors pre and post COVID, you can tell this dip in activity was all around March 11th in 2020, empowering patients really to collect information on their own health and participate in research directly. This is part of the Apple Heart and Movement study, which again, we were able to attract as a result of that initial seed award in the BRI Shark Tank. And then ultimately, one of the other elements we recognized in this program, and we were able fortunately to fund, is the ability to train the workforce of the future, to enable people across the entire spectrum, including patients, to train and understand artificial intelligence and biology in ways that allow them to participate really actively in the research and care delivery process. I would be remiss if I didn't thank, obviously, the Shark Tank judges that gave me this original award, all of the individuals who are on this slide, but none less than Rahul Deo, who's been a really remarkable partner in this. And then some additional funding, very early stages from NIH, from Eamon Corey and from Steve Mara, that has helped us really drive a lot of this work forward. Thank you.
Great, Callum, that was spectacular. And just to see the amazing progress from that Shark Tank sort of became the kind of apex predator project here that has, rightly so, gotten just tremendous funding. We have only about 60 seconds left. There was a good question in the Q&A from Dr. Hada. Maybe I'll take a slight spin on it. It had to do with the specific inputs and outputs for the model for AI-based approach. But maybe in 60 seconds to say, with all this data and with using AI and machine learning, in 60 seconds, sort of, what's the takeaway for people thinking, is AI or machine learning going to sort of solve everything in the future? What's the takeaway you would give to people for that?
I think the biggest single takeaway for AI, in my view, is that it's not a one-shot deal. It's a continuous optimization. It can only be done in parallel and in conjunction with testing the output of the original AI models that you've developed. And so I think if you look at it in other industries, the way it's used is really to do that. It's to take a process and begin to systematically optimize it by understanding how it responds to different challenges. And what we've done is really introduce those challenges very early on in a very clinically relevant and broadly available fashion. And so to directly address Dr. Hada's question, we use traditional clinical inputs and we use traditional clinical outcomes. We built essentially raw feeds from the electronic data warehouse across the Mass General Brigham system. So it's trained against real world outcomes in our system. So yeah.
Well, that's fabulous. Okay, we're at the end of our 45 minutes. I want to thank Vic, Jean, and Callum again. It looks like we bet well on all of you to give you the funding. You guys have done tremendous work for really important disease states.